Hello, my name is Meredith Sanders, and this is my Lab 3 3D model of my protein for Intro to Cell BIOL 2440 with Dr. Hudson. So I got the honor of being able to be assigned the protein Porin. So I have a 3D model going here. It's just kind of spinning around in slow motion, and then I'm going to get to this, which is just a simple explanation of the 4 different structures, which I think it's drawn out in a way that's explained well. So I'll make sure to include that later. So I feel like we should just have like a quick summary of what Porin protein is. A Porin protein is actually super fascinating, and I'm really glad I got it. So the function of a Porin protein is kind of to act as a channel. It acts as a channel for molecules to be able to come in and come out and like diffuse. So what's pretty fascinating is how Porin proteins are channels that are specific to the molecules, right? So depending on the molecules, the channel will be smaller, bigger, all different kind of stuff. So Porin proteins are pretty specific. They're not just barrels, you know, and that's really cool. And so mainly though, they transport hydrophilic, meaning they like water, hydrophilic molecules across the membrane. And Porin proteins are found in eukaryotes, gram-negative bacteria and gram-positive bacteria, but mainly gram-negative. And just a shout out to the man who discovered this back in 1970, 73-ish. It was Hiroshi Nikaido, or Nikaido, however you say his name. So yeah, and so we're kind of just discussing the 4 different structures and being able to identify them based on looking at a model like this. So there are 4 protein-like structures, and most proteins actually have the primary, secondary, and tertiary, but some really don't have the quaternary. But surprisingly, well, we'll get into that. So the primary structure, we can think about it as just like a bunch of it's a polypeptide chain with a bunch of amino acids, and Dr. Hudson explained it in a way that was really helpful for me in that it's kind of like beads in a necklace, right? So they're just all connected to each other in this long chain. And it's displayed here, and I'll make sure that will be at the beginning and the end of the video so that you can just look closer at it. That's the primary, and the primary structure, yeah, okay, so yeah. And then the secondary structure is more 3D. It's than the primary. It's the one that has 2 types. It has alpha helixes, which are kind of, as you see here, there's times where there are actual spirals. So like, okay, for instance, this is the beta sheets, and those are the ones that kind of, they loop, right? So it kind of looks like a weaved basket, but the ones that are more coily, kind of like, you know, like my hair, like a ribbon, those are the alpha helixes. And the structure of a secondary structure, it's made out of folded structures that form polypeptides, and those are stabilized by hydrogen bonds. So we have a lot obviously going on, a lot of secondary structure that you can just automatically see with the Porin protein. So then the tertiary structure is number 3. It definitely has a 3D look to it. That's it has 1 polypeptide chain, which they call like the backbone, and it has several secondary structures in it. So what's interesting is they all kind of form onto, like, they're, so like primary structures is involved with secondary, which is involved with tertiary kind of thing. So because like if you look at these different, all these like plated, the beta sheets, those have the peptide chains in them, right? They have that beaded necklace going through them. So it doesn't display that, but we kind of just know that, right? Yeah, so that's what that is. And then the 4th one is the quaternary structure, and that's basically like the cluster. It's the cluster of all the protein shapes into this like final structure. It's stabilized by hydrogen bonding, salt bridges, and disulfide bonds. And yeah, so basically, like, the structures can be simplified by saying, like, the amino acid sequence, the characteristics of many proteins, the 3D structure, and then the 3D structure of numerous subunits. That's like the primary, secondary, tertiary, quaternary. Yeah, so as we look here, we know that we have the primary structure. We can see the secondary structure for sure. We have this barrel going on, and like I said, like the weave baskets, that kind of, yeah, and it creates, like, there's 3, so this is 1, 2, 3, all the different colors represent a different, like, barrel, a different channel. And the most obvious is that it has tertiary structure because it's 3D, and this is actually technically 3. Like a Porin protein is just like 1, so this is 3 that have, like, been put together. But yeah, the channels, so whenever it comes back around, whenever it looks like this, that's the channel for the molecules to be able to diffuse and move around. And so yeah, it's super fascinating, the Porin protein. I'm super excited to be able to research more and do the paper and stuff like that. But yeah, that's my 3D model, and stick around to the end, and now you'll get a closer view of the images I kind of explained with the structures. Thank you.